my screen. I hope that was a useful little break. And so we welcome Amanda. Thank you for coming and dedicated your time to us today. You have the floor.
Okay, I have unmuted myself. Hi, everyone. Thank you for coming to this workshop. This is such an exciting time, and just think by how engaging this is in the number of you guys just getting up during the summer and doing this work. This is so awesome and so encouraging. To be honest, I don't know. I even was thinking in the lines of being a scientist. So my journey began a little different. So it started in South Africa. As a little girl, I always thought I wanted to be an economist. I just really loved economics and I got it. And it was just awesome until we had in our school, we had these nurses come and they started talking to us about this virus that was going around, and it had a high mortality rate of the population in South Africa, which was kind of a little interesting. But then I wanted to know more about this virus because the way they were explaining it, and I just back then we were not well versed about it, and you were just told that if you see someone injured, you had to take plastic bags, wrap your hands around it, and then you could help them, but you couldn't just go touch people. So it kind of, because of the information that was put out there, there was a little bit of hysteria, and we didn't understand HIV well until I became a little older. And when I was in high school, I started to understand in basic biology classes how viruses work, and how we could actually protect ourselves from it. And so that's how my story began. And I always just knew I wanted to be a wet lab scientist. I came to America and then I studied biotechnology. And for a while, I thought I was just going to be into protein purifications. But life had a different journey for me. I got my first job working on campus, and I was working in a microbiology lab. So I had two campus jobs, one to work in a microbiology lab, the other I was working in a chemistry lab, and I was just more inclined into the biology. And then I got an internship at the Howard Hughes Medical Institute, and then that's where my love for neuroscience began. And so I was at Genelia Farms. After that, I moved on from there and I went to graduate school. And when I was doing my master's degree, I started to learn about bioinformatics, which I had no clue what it was about. It was just at the time where the buzz of CRISPR was just coming around. And then one of my professors said to me, do you know that the first CRISPR experiment was a bioinformatics experiment? And I was like, wait, what? And he's like, no, actually the guy who discovered the short palindromic repeats used a word processor off the coast of Spain. And that's where I was like, oh my goodness, yes, I'm hooked. That's actually how we put proteins together as well, just by using letters and a word processor. And here I am still working at the same lab as a bioinformatician. So I am now the lead. I used to be the co-lead of this project called OncoMX with one of our master's candidate, PhD candidates, who's now moved on and is working at the NIH. And now I'm the lead of this project called OncoMX, which is onco for cancer. M stands for mutation, X for expression. So in this database, we collect all cancer-centric biomarkers and their metadata. And the metadata that we collect are for expression data, mutation data for healthy expression and disease expression, because we use that to compare the data sets. One thing that I would like to point out, all the data sets that we see here are for the expression and the mutation, all comes from RNA-seq data. So I was super excited just hearing the previous talks, and the theme of this week being RNA-seq, and everybody just talking about RNA-seq. So we put together this project. It was funded. It was very database centric. So what we did is we brought together all databases and we standardized their data into one resource, and then we make it available for various people, for people such as you. If you have a project that you would like to write about a specific gene, you can bring your gene, put it into our portal, into a search button, and then you can see all the various data points for the specific gene that you would like to see, as I will demonstrate later for you guys. Okay, so what kind of data is there? That's what I'll go over a little bit, how you can explore it, how can users search for a specific gene, and what are the future. And just to go over, the reason why I'm here is because you are the future of such resources today. And I actually I coordinate a big cohort of high schoolers, which we started during the pandemic to collect biomarkers. So they collected COVID-19 biomarkers. We have a publication out there, and we gave our high schoolers authorship because they've done such tremendous hard work on it. And so we are using this same model now into the cancer space. And I'll tell you how you can be part of this, and you can work on it. Right now, we in the second cycle, we run 6-week cycles. So each cycle is equal to 2 weeks. So we in our second cycle of our 6 weeks. And if you guys would like to join now, you may go ahead and join and just stay with us through the summer, or you may stay throughout the year and just keep on curating and submitting biomarkers. And obviously, we will reward you with, we acknowledge you in the papers, and also the biomarkers that you submit. We actually attribute it to the person who submitted that biomarker. But a little bit about that in a bit. Do you guys have any questions? I know that sometimes I talk so fast that you guys can catch up, or I can't catch my own breath. So I'll pause for a few questions if you have any. Okay, can somebody just tell me that you at least can understand me and you can hear me? I just need one little voice out there.
Yeah, we can hear you clearly.
Yay! I love that. Okay, so next. So as I said before, so quickly, I just want to go over this. It's major databases that we have in our resource. So the two core databases were created by us at the GW lab. And those are called the mutation datasets and the differential expression datasets. But then we also have normal expression, or what we call healthy tissue expression. And that is created by a lab at the Swiss Institute of Bioinformatics. I was super excited. I believe we have someone in the UK. We collaborate with a lot of European institutions. And so I'll tell you a bit about it later if you are interested in that. And then we have literature mining search tools. And these tools are applied to search for mutation and expression in respect of cancers in various public databases, mostly in PubMed. And then of course we have the biomarker, the core biomarker data sets. So we are now working on the third one where you guys come in, but the first one was from EDRN, which is the early early disease research network, and also from the FDA, which is the Food and Drug Administration. Okay. In addition to our core data sets that we put together, we had to retrieve the data that we have in our core data sets from various other data sets which are well established. And so we got them from the NIH, the ICGC, the Sanger Institute, and various other smaller databases. We cleaned up this data. So the one good thing about bioinformatics, so opportunities are endless and it is so broad. As a bioinformatician, you can work in building databases. As a bioinformatician, you can work in being a data wrangler. As a bioinformatician, you can work as being a data analyst. That's more what I do. I do data analytics, but also then harmonize it, ontologize it, and bring it into bigger databases. So there's a lot of different paths that you can follow as you are interested in. Okay, so then when we get all these data sets from various databases out there, we need to bring it together so that somebody can find it useful. And it's not just pieces of data that is not harmonized or they cannot work together. So we need to find various ways to do so. And how we do that, we use IDs. So I've heard NCBI being mentioned earlier on in OMEM. So we use those kind of well-established institutions' IDs to harmonize our data. And one of our major ID that we use is UniProt KB, and for our genes, we use Ensemble. We also have RefSeq IDs, and then we have another layer of IDs, which is the biomarker IDs, and those are the IDs that we assign for our resource. And then there are various ontologies to harmonize bioinformatics data and bring together such big data bases. We also have other little projects research projects or companies in the sense, for a lack of a better word, to have you understand. And what they do, their sole job is to take all the data that various universities, various labs, various projects put together, and they find a single common entity to harmonize this data set, and they apply an ID to it. So let's say for tissues in our body, we can extract various tissues from our fingers, from our face, anywhere in our bodies, but then we need to give these tissues names. But because there's so many people working in research, we also need to find a way that everybody knows what specific spot you are talking about. So then we have this thing called an Uberon ID, which takes a cross-section of a tissue, which is called the Uberon anatomical entity, and they give it an ID and they give it a name. So when you put it in a database, you're able to identify it. When it comes to disease, we have various cancers. One specific one that I like to use is colorectal cancer. People sometimes can just go with colon cancer or just rectal cancer, but then there is something else that is colorectal cancer, which is part of the colon and the rectum. And so we need to find a way to harmonize it, to call it that. And so when somebody's working on a specific tissue site or a disease, they find the exact data that they are using. So that's just a quick snapshot of what we are doing. I'll show you guys a use case later on how you can do a search, especially for you guys who are upcoming and would like to just take a stroll through databases and just understand how various cancers come together, or their metadata or mutations and expression work in genes. So you can just type your gene name. If you have a favorite gene, you can type it in here, and it will give you all the relevant data that you can read about. And so you can also search for a specific biomarker if you would like to look at a biomarker for a specific disease and to see how that marker is actually being represented in the tissue type or the disease. And here you can see if it's an EDRM biomarker, what they use it for. You can also see if this biomarker is commercially available and if it's approved by the FDA and for what it is approved for. So it could be approved for a prognosis. It could be approved for as a risk factor for a disease. It could be approved to diagnose a disease. There's so many best biomarker categories that we go through to see what specific biomarkers are used for. So this is the other side. So the previous one that I was showing was EDRN biomarkers. And here we have for the specific type of cancer that is my cancer of interest is lung cancer. So we have a 28 FDA approved biomarkers. And then we also have, where now I'm gonna go into where we need you. We have these curated biomarkers in OncoMX. A little earlier, I mentioned that we did COVID-19 biomarkers, and this job was done solely by our crowdsourced interns who worked so hard to put together a very beautiful COVID-19 data set. And now we have this OncoMX biomarkers, which is also using the same data model and mechanism to collect these biomarkers. The one thing that is fascinating about this project is it is a project that helps high school students or even undergrads just to catch up with literature in the biomarker space. So it puts you in a position where you don't need to go and download additional software on your computers. You can do this anywhere, anytime. All you need to do is have access to the internet, start reading papers, and then you submit your biomarkers as you find them in the papers. It goes through a Google form, which I'll show you guys in a little bit. We get the data. My colleague adds diagnostic codes to the data set, which are these diagnostic codes are called LOIN codes, which if you become part of the project, we go through rigorous training to just explain the various part of entities for these biomarkers and what all these different codes does. So this information is useful to researchers, it's useful for clinicians, it's useful to the public in general, just to have an overview. Do you guys have any questions before I proceed? Nope. Okay, then I'll go to the next slide. Okay, so the work that you guys give to us or the biomarkers gets cleaned up, and once all the biomarkers are reviewed and QC'd or quality controlled, we then put them here in the biomarker portal, which is on the front end. And from there, other people can now come and search the curated OncoMX biomarkers. Okay, so this is the data model, which I will also demonstrate when I start getting to the form. The various things that you are looking for when you do your search on the internet is to find the biomarker ID is left alone. You're not responsible for that, but an assessed biomarker will explain that, what it is. We need the biomarker name. We need the specimen type, so various specimens from where the biomarkers can be collected from. Usually papers will write, and sometimes papers are too ambiguous and they don't have the precise specimen type of the biomarker. What you do, you just leave your little note to say, hey, they did not have the specimen for, so I don't know if this is coming from serum or blood, or if this is a solid tissue biomarker, and then we work with you through that. And of course, then we also assess the disease type. So the biomarkers of interest that we are doing is cancer specific. So we have 15 various cancers that we are looking at. And so then you will place in that information and we will collect it. And then if you, once you send us your biomarker, we put it on the site. And if you go and search, let's say you don't know your biomarker ID, but you're like, oh, by the way, last week I submitted a biomarker for lung cancer. So you just type in lung and you click on there and you will see your biomarker with your name on. Or you say, I don't remember what the cancer was, but this is the name of my biomarker. I submitted a biomarker which was called interleukin 2 or 6 or D-dimer. And you type in and then you can see, oh, yes, you did. And it was for this cancer. And this is what the biomarker does in that specific cancer. Okay. So I'm gonna paste a quick link in the chat when we're done, or I'll put it on Slack. But this is how where the biomarker curation project starts. We have a Google Doc that I will give you access to. There's a lot of information on a Google Doc and how you can curate your biomarkers. And if you get stuck, we will add you to our Slack group and you can talk to us and we can guide you further with the biomarkers. Before I go there, let me go to the doc. Forgive my screen in the background. So this is the Google Doc that I will give you access to. It's tiny URL, OncoMX biomarkers, and my PI is Dr. Mazunda. The team leads for these cancer biomarkers are Asia Joseph and Dan Lyman. So I get my orders from them. So we have a biomarker registration form. This is where you'll go first and you'll click on this biomarker registration form. And when you click on this form, it will tell you a little bit about what the OncoMX biomarkers are and what we're looking for. We want to know more about you as well, because what we have noticed is we find such a diverse group of students and we try to develop your skills as best as we can. We have students who are more inclined into programming. So we find projects in machine learning and artificial intelligence. So our lab has 6 different divisions of projects. So my project is cancer biomarkers, but if we see your skills are in line with another project, such as we have the gut microbiome, which is AI or artificial intelligence and machine learning, then we put you together with that group and you can intern with them. It's all online. All you need is just your computer and everybody walks you through what you need to do. So, but everybody starts kind of at the biomarker project. So you tell us what your school, your university, your high school, or any institution you are in. We have various collaborators right now from various parts of Africa, Europe, India, and the United States. Just how you heard about the project, and in the comment section, you can actually tell us what you would like to get out of this and why you are doing this. And there's 4 of us who works on this, and we try to really get in touch with all our interns and make sure that we develop you. And when you eventually go out, you have the skills that you need to go to college or continue going through high school.
Yeah, I have a quick question. Is there a minimum age for that, or is it open to everybody?
So we've had 8th graders who were just fantastic. So we really don't. We just feel that we have to be so inclusive that anybody who can do the work and shows interest and would like to receive the training, we are here to help you. So yeah.
And is there some kind of a prerequisite? Oh, Jenny, go ahead.
Oh, it's not that important. You said there's little programs for like AI and other things, and like if you sign up for this, they'll send you to those programs. So do you have to sign up to this link, or is there like a link that you can send for those type of programs?
You can sign up to this link, and if you are interested into artificial intelligence, we can put you. So my other colleague, Dr. Keeney, runs the AI program, and then Stephanie runs the machine learning in MATLAB for gut microbiome. So yes, we can put you.
Okay. And
Yeah.
we can go for the internship this summer. It doesn't have to be next summer.
You can still come this summer, and we try to design our internship so that our students can also stay throughout the year. We've had students who just stayed with us. They joined us in 10th grade and they stayed with us until they graduated.
Wow.
We have, and you can go on and off. We've had students who would say, okay, my exams are coming up, so I'm not going to work from April to about June, the end of June, then I will come back and I will curate. We like to work with students. We like to stay engaged with students. So the only thing that I will say is communication. Don't just drift off, because this is kind of, we teach professionalism, and this is how you're going to go into the world. So while we're providing you with the hard skills, we also try to give some soft skills on how you act when you go to work, and if you cannot do something, how you communicate ahead of time, and we also don't put the pressure on, oh, you didn't submit a biomarker. It's all voluntary. If you can submit it, you please submit. If you can't submit it, nobody's going to come and say, why didn't you submit? Daniel, do you have a question? Daniel's hand is raised.
Are the internships in person or online?
They are online. And so I love young people, and I love just seeing my students. So I try to arrange a big event in the summer to have everyone come, even if it's for one day. We try to go to the university and get a little bit where we can see if we can help, because I don't think it's fair to say to students, oh, come visit us here, but we can't provide you with a little bit of accommodation or something like that. I just think it's sometimes good to do face-to-face, but because we bioinformatics, we're all online. And if you want to come to GW, so we set up a whole tour with the department, MIPI, in various labs at GW. And you can just like walk around and you can come see, you can work for a week at GW, whichever you want. And if you just want to work on a full project, even if it's a wet lab project, we put you in touch with another PI in various, everything in the Department of Biochemistry. Doesn't have to be cancer-centric. It can be any tropical diseases. And then you can work with them for a summer, or however you guys make the arrangements.
Yeah, and I also wanted to ask if you said it's there are some compensation, right?
We don't promise it up front, because we don't know how much we get from the department. It all goes within that year's budget. So
Okay.
okay,
but the kids know in advance whether they're going to be paid or not.
always in advance. Yes.
Okay.
Yeah.
And do you have to be in the US?
No.
Okay.
Wherever in the world. One of our super reviewers, although he's coming now to join our master's program at GW, he is in Ghana, and he just spoke, and so with my PI, and then my PI got him, because this person is super, super talented. And so they got him to work to see if there's some scholarship funds and things like that. So the more you get involved in various tasks and projects, you are also exposing yourself to getting more funding to take your education further.
Yes.
Do you
Jenny's asking, oh, go ahead, Jenny. You can talk.
I was just like about to say, do you need any prior requirements or experience in engineering and bioinformatics?
Actually, no. And this is the fun part that I actually like our lab. Our lab is called the Hive Lab. So I'm going to give you a quick overview about myself. So when I started, I came from a wet lab background, you know, and I didn't have bioinformatics experience. And I was just thrown into this project. I was a master's student and I had to do this project on this highly technical project, which is like a computational biology project. And Huntington disease was a disease that I worked on, and it was a standardization project. And we just received IEEE standardization in 2019, which is a big, big deal because now they recognize it as a standard and it's called biocompute. But we teach all these skills as you go along. You don't have to have it. All that we ask for, have an interest, love what you do, and we'll go with you every step of the way to where you need to be.
Okay, thank you.
Yeah. Yeah. So these are just if you guys I'll put the link in the Slack if you guys are interested. These are just the links to tell you what you can find, where you can find various other things. You can look at some of our older meetings and see what we discuss during our meetings. It's all virtual and online. This is available for the public to see. CC by 4, all the work that we put on in the cancer biomarkers are CC by 4 licensing, meaning that anybody can download it and you can work on it, you can cite it and whatever. All that we ask is that you please just cite that you got it from our source, but it's open to the public for analysis. Our project goal, then we talk about our project meetings. So here's where it becomes a little tricky, especially with our younger population or interns. If you have school and classes, then we don't, in the summer, yeah, you can make it every other Monday to attend a 10 a.m. meeting. But when once school starts, we can't expect that from you. All that we expect from you is to like send a quick Slack, hey, I am still part of this. I am reading this paper and it's taking me 2 weeks. Or look at this paper that I found, but I'm struggling to find the biomarker in it. Can you please help me? So we will actually help you break down that type of information as well. So now that you have found your biomarker and the paper that you are interested in, then you go into here and we now start to collect the data. Oh, sorry, this is a different one. It's not the one that I wanted to show. Let me just go out of this. I wanted to go to the collection page, and I think unfortunately I did not link it here, but I'll show you where to go to the biomarker collection page. These are all our biomarker lists. So here's the most important page. Once you're part of the team, now you have to submit your biomarkers. And a lot of the things that I'm going to show you here are things that you guys went through over throughout this week, because it's all about understanding and learning databases and know what to get from which database. So if you read about a specific gene or a protein or a molecule that is used as in a biomarker, first thing that we ask you is go see if you can go into UniProt, so UniProt KB, and you put in the specific gene. So I have UniProt here, and let's say, for instance, okay, I'm reading a paper and my paper has EGFR. So then I just go EGF. EGFR is the gene that I'm interested in. And then I look at here, oh, there is actually a lot of human EGFR. So all the biomarkers we're accepting are for humans. So we want you to please pay attention that it says human. And then you say, okay, yeah, EGFR is the epidermal growth factor receptor. Very interesting. And what is the species? This is the quickest thing that you go and you go, okay, it is for the human. And here's another quick tip that I will show you guys. If it's not a human gene, then the gene name is written with one uppercase letter, but the rest is lowercase. But if it's human, it's all uppercase because it's HENC, which we'll teach you guys a little bit later as well. So that also just helps you when you see a data source quickly, you are able to deduce without even having to dive in too deep. You can also say, oh, this is not human. Look at the way this gene is written. Oh, this is human. So just small little tricks like that. And from there, you're gonna extract the protein ID. So you just copy and paste the protein ID, and then you put it back. Hold on. And then you put it back in here. Not here, you put it back in the form, in the biomarker form. Which one was it? Nope, wrong one. Yeah, there we go. And so you just put the UniProt ID here. So here could be P we show you exactly how you want it to be. Then the biomarker that you're looking for, that you were reading was EGFR. Then I just type there. It doesn't matter. Here, I don't care if you do the uppercase or lowercase, because I'm gonna curate it anyway. But if you wanna make our jobs a little easier, you can just put it in cap letters, EGFR. Then it's asking you, okay, what does EGFR do? So if you read in the paper, you say, okay, they say in non-small lung cancer, EGFR expression is elevated, or EGFR expression is higher, or something like that. Then you just write whatever you say, like elevated EGFR expression levels or something like that. You know, you just make it up. Then this is the very tricky part. This is the NIH best biomarker types. So they tell you in various categories that you can rank biomarkers for. So here you kind of have to think and make your best judgment and your best guess. We go through it and we will show you as time goes by, you will not get it right the first time. I honestly did not get it right the first time, and I still struggle through some of them, where you kind of have to figure out, is this biomarker a monitoring biomarker? What is a monitoring biomarker? Okay, they have a patient here. This patient has cancer. So if they have too much of this, is their cancer getting worse? So you can see if it's monitoring or prognostic. If this biomarker is getting less, is it becoming better? But then it gets a little higher later. So they can fall under prognostic to risk. If they have this, so risk falls more on mutation, on biomarkers that is shown with mutations to say, okay, if you have, let's say, a BRCA1 mutation, you have the risk of breast cancer. Then we have a diagnosis where the paper sometimes will say, this biomarker will be used as a diagnostic. The patients that we saw had this expression or mutation or lack of this expression that were diagnosed by this type of cancer. That's how we do it. And we have safety, which we don't find a lot in response biomarkers. We actually haven't found any of those in our resources yet. So that is just also a lesson learned for us that we need to refine pipelines, and also maybe in healthcare as a whole. How do we quantify response and safety biomarkers? And then we have predictive biomarkers when you can make a prediction of a diagnosis. due to the biomarker. Then we have the specimen type. The specimen type is how they extracted the biomarker. Sometimes they get like a blood sample from your arm. Sometimes it's from a tissue or biopsy, a solid tumor that they get it from. So just read how they got the sample that they are testing. And then you can put there it's blood, and then you just put in the code for the specimen type. And then we have a disease. So in this case, this is a COVID-19 disease ontology, but because we have a list of very specific cancers. And I will just say to you, other than the biomarkers, everything else, we have a very narrow list for you to choose from. So you're not just flying in blind, you just go, okay, I'm reading this and it corresponds closely to this entity. And then you just choose the entity. And we do a pretty good job that we actually go through the papers and we use it as a teaching and training mechanism that we go back to our curators and we're like, hey, do you want to talk about this? Sometimes I will write you an email to break it down. Sometimes I will write you a Slack, whatever you're interested in. And then one very important aspect. Do I still have time? Am I running out of time? Sorry, Stephanie. Do I still have time?
Sorry,
Okay.
I'm muted. I thought I was
Sorry.
muted. Yeah, so you have time. You have time.
Okay. I'm sorry, guys. This is super technical, but once you like dive into it, it's very enjoyable and not so technical. It's just work with big structures of data. One of the things that hopefully I'll find in this group is someone who can do phenomenal visualizations and can maybe find a not so boring way to show this. And that's also a job that plays a lot in bioinformatics, by the way, visualizations. If you like art and drawing and showing things, you will be surprised what you can do with that. One important aspect in the paper that you found, we need you to take a sentence out that writes about this biomarker to say this biomarker does this. So here's an example. Serial measurement of circulating IL-6 level may be important in identifying disease progressions among COVID-19 infected patients. So it will say biomarker, we see that biomarker X or whatever your biomarker is implicated in patients who have cancer, lung cancer, and it is upregulated, downregulated, or it's a mutation, or these levels are just higher in the case of D-dimer and various other cancers. And then here you can just free write your notes. It can be whatever you want to write. If you want to tell us something interesting about this paper or something we should know about this paper, please go ahead and just put it in the note section. Or if you even have a question, put it in the note section and we will address it. Most importantly, we need your digital footprint in research. So we have all our interns set up an ORCID ID. When we write a paper or somebody else write a paper from using our resource, we include you in our papers and we put your ORCID ID. So you can actually go when you on your college application or your CV and say, hey, I've contributed on this paper. And if you click on a paper, you can actually see my ORCID ID is in that paper. And it also will link automatically to ORCID. So ORCID will like notify you and say, hey, the first name that I'm seeing, I'm just gonna say Daniel Webster. The cancer biomarker team published a paper. You helped on that paper and they have acknowledged you on that paper. So that publications is kind of like currency in research. And I think if you're gonna go through academia, you guys have such a wonderful future ahead of you. It is good to have a little head start to say, okay, you are contributing to major projects. It's making a difference in this world. So that's all that I have. you guys have questions for me, and I'm sorry for going on and on about really flat, boring pages.
Important. I think Daniel had a hand raised.
Oh, hi, Daniel.
No, I'm okay now.
Oh, you okay? And if you guys just like sign in, you guys can just ask me questions there. I'll put this in the link in case you want to be in contact with us. You can just let us know.
We can
And
put the
we'll
whole
always be
page
there.
on Slack, right?
Oh,
I
yeah.
said you can put all
do this.
the whole set of link on Slack.
Okay, I will do that. So I'll put that. And then you can just see these are the people who participate. The PI is Dr. Mazunda, and then we have Asia, and who's the co-lead. She's currently our master student. And so she is co-leading this with Dr. Lineman, who's an ontology expert. And then I do the fun job. I get to work with you guys and do the analysis. So the fun part. And then we have Ned Colley, who's totally awesome. And if you guys want to learn just doing some a few backend stuff, like even just getting the using command line to get a data set up, he gives like little fun tutorials. But yeah, you kind of get a little by little dive into bioinformatics.
Mhm.
Yeah. So that's it.
Thank you. Anybody else has a question?
No, that is cool.
No? Yeah.